Hello. Thank you so much for inviting me and giving me this opportunity to talk about our epigenetic clock studies in companion animals. So my name is Steve Horvath. I work as a principal investigator at Altos Labs. I declare a couple of interests. In particular, I founded a non-profit epigenetic clock development foundation that offers epigenetic clock testing to the community. My research is motivated by the challenge of measuring biological age. Many people are working on this task, and people come at this question from different angles. On the right-hand side, you see various clinical manifestations of aging, frailty, muscle loss, lung disease, cognitive decline. And one can, in certain ways, measure the biologic age using these clinical manifestations. And on the top, you see the bullet of cellular changes, tissue pathology. So a pathologist can look at tissues and again assess organ dysfunction. In my lab, we look at the lower bullet, which are genomic data. My entire talk will be about so-called methylation data, but I want to mention that there are many other genomic data sources, gene expression data, proteomics data, metabolomics data, glycome data. And each data source I list here has led to different ways of measuring age. Now, what all of these methods have in common is that they arrive at biologic age estimates that are strongly correlated with chronologic age. Otherwise, it is not a biologic age measurement. And so, as I mentioned, my lab works on methylation, but I want to mention that it's been long recognized that methylation changes with age. I think the first publication came from a Russian investigator, Berdyshev, who looked at the spawning humpback salmon and noticed that globally methylation decreases with age. But this was 1967, where they looked at global levels. Now we are in the era of sequencing and methylation arrays, and we can look at individual cytosines. So as you know, the DNA molecule has 4 letters. One letter is the cytosine, and cytosines can be chemically modified by the addition of a methyl group. And statistically speaking, you get an Excel sheet where the columns are different people of different ages. The rows will be tens of thousands, if not hundreds of thousands of these cytosines. And the values are proportions indicating the proportions of DNA molecules that are methylated at a particular location. And we define epigenetic clocks as a multivariate regression model whose dependent variable is chronologic age, and then the covariates are these cytosines, the methylation. And the first epigenetic clock was published by our team at UCLA for human saliva samples, 2011. Shortly thereafter, there were a couple of papers on blood-based methylation predictors of age. In 2013, I published the so-called pan-tissue clock that measures age in all nucleated cells. All cells of the human body that contain DNA lend themselves for an age estimate. Now, later, we aimed to develop human mortality risk predictors. And that gave rise to the second generation epigenetic clocks, PhenoAge, GrimAge. So again, these clocks don't attempt to measure chronologic age. It's really mortality risk. And lately, we're in the third iteration of clock building. We are developing epigenetic clocks that apply to multiple mammalian species. And that's very relevant perhaps for the health of companion animals, and I'll focus a lot on that. So if you're a statistician, you will recognize that we're dealing here with machine learning, regression analysis, for example. So there are many ways to build such clocks. And by now, we have dozens and dozens of different clocks. Here I show you the human pan tissue clock. Each sample is colored by tissue type. And you see a very strong correlation, which actually starts at birth. So from age 0 all the way to 100 years, we see very good accuracy. But you know, if you look deeper at the regression model, what is hidden there is that we are using a non-linear transformation of age. Here I show you on the y-axis, in essence, a mean methylation value. On the x-axis, chronologic age. And you see that during human development from age 0 to age 20, there's this non-linear relationship. There's a fast increase in methylation during postnatal development. And then later in life, after age 18 or so, it levels off and you get a linear relationship between methylation and age for these biomarkers. And I wanted to mention this nonlinear relationship to you because we actually use a very similar transformation when we build mammalian clocks, clocks that apply to all mammalian species. But before I move on, I want to point out a deep biologic insight based on this kind of behavior of methylation, which is changes during development correspond also largely to changes at old age. And that's really one of the messages of epigenetic clocks, that developmental processes that are important and useful early on may at some point go awry and then be associated with aging and organ dysfunction. So epigenetic clocks link development and aging. So here I show you a cartoon of the same curve that you just saw. The blue line represents epigenetic age. And the intuition is, of course, that if you lie above this line, if your methylation age is older than it should be, maybe it means that you're at a higher risk of morbidity or mortality. Conversely, if your methylation age estimate lies below that line, then one can hypothesize that maybe you have a lower risk of morbidity. And this is a nice hypothesis, but it really required tremendous effort by the research community to establish that and to really quantify it. And that has been done largely by epidemiology. Now, what these studies have shown that, yes, these first generation clocks, they do predict mortality risk, but weakly. And therefore, we and others developed so-called second generation clocks. So these are now human mortality risk predictors that want to predict time to death based on retrospective cohort data, where, for example, people collected blood samples in the 1990s And we have now 30 or 40 years of follow-up information. So we know whether the person is still alive or if they died, what they died from. And so then again, it's a regression analysis, for example, Cox regression analysis, time to death. And so here I show you our best mortality risk estimator named GRIM-H, named after the Grim Reaper. And this estimator relates to anything that you would expect it to relate to. So it strongly relates to smoking, body mass index. If you have fatty liver disease, you have higher values of GRIM-H. Conversely, vegetable consumption is protective. So all of these studies have been done in many different human epidemiologic cohorts. Here I show you just one example. This is from a Scottish cohort, the Lothian birth cohort, where the authors showed that if you are in the bottom quartile of GRIM-H, your survival probability is higher than if you're at the top quartile. The top quartile, this is a blue curve, and you see the proportion of people who survive after following them for 10 years or so is really greatly diminished. And here I did the same thing for many other epidemiologic cohorts, the Jackson Hart study. So now we compare the bottom 20% fastest agers according to GRIM-H to the top 20% agers. And you always see a pretty good separation. By the way, the analysis is stratified by sex. It's, as you know, women live longer than men. So, but any way of stratifying the data will show a separation. For example, you could say, evaluate GRIM-H in female smokers or in male non-smokers, and GRIM-H always helps to stratify mortality risk. GRIM-H and these kinds of biomarkers are being used in human clinical trials. Here I show you just one slide on a study by James Clement, who looked at a particular treatment using so-called umbilical cord plasma concentrate. And this was injected in 18 people. The mean age was 74. So these people received an injection of this young plasma for 10 times over a 10-week period. And then, yes, it did rejuvenate GRIM-H by a little bit less than 1 year, 0.8 years. It's not dramatic, weak effect, you know. And so clearly we want better treatments, you know. We would like to find treatments that have a much stronger effect on GRIM-H. Here I want to convince you that lifestyle matters for humans, of course. You see strong relation. The top row shows you mean carotenoid levels. Fundamentally, it measures how many vegetables you eat. And you see a p-value 10 to the minus 39, a correlation minus 0.26. So to me, as a statistician, that's a strong correlation. And yes, at lunch and dinner, go for the vegetables. In the middle panel, you see C-reactive protein, a marker of inflammation, is positively associated with GRIM-H. And then at the bottom, you see things like smoking status, very strong effect, negative effect. And so that smoking has a very strong effect by construction of GRIM-H. We constructed it so that it would pick up smoking. So, but why do I show all of that? I show it because we really have wonderful epigenetic clocks in humans. But what about developing such epigenetic clocks for companion animals? And then I should pose here some open research questions. Does a healthy lifestyle in animals correlate with reduced epigenetic age in these animals. And I personally don't have any data on that. Maybe some of you will be working on it. And then the next question is, well, what constitutes a healthy lifestyle for companion animals? And I pose that question. I bet all of you have an answer, but what I can tell you, in humans, we know that based on 50 years of epidemiologic research. We know smoking is bad for you. We know obesity is bad for you. So we really understand quite well lifestyle effects on humans. But maybe these types of epigenetic clocks can actually help us identify what kind of lifestyle interventions, what diets have a benefit in dogs or cats. Now, to facilitate this kind of research, we do need to design mammalian analogs of these epigenetic clocks. We need a mammalian analog of a pan tissue clock, and of a Grimm-age clock. And I want to briefly mention a technical challenge here. So past measurement platforms that were designed for humans, so the so-called human Illumina Infinium array, really doesn't work for rats and dogs and cats. And so we wanted to design a new so-called mammalian methylation array platform that profiles the cytosines in highly conserved stretches of DNA. So we wanted an array that applies to all mammalian species, but focuses on conserved DNA sequences. And this resulting mammalian array was designed to tolerate mutations, right? So a limited set of mutations in DNA sequences can be tolerated without affecting the measurement of the methylation. And and I wanted to give you a sense of how many cytosines can be profiled in different species. So here I show you different taxonomic orders. Top one is primates. So in primates, I want to say we have maybe 34,000 probes. Then you see rodents, far fewer, 28,000. Bats, similar number. And then at the bottom, we have the so-called monotremata. These are species like platypus, which are these egg-laying mammalian species, and there we have maybe 10,000 cytosines. So in any event, the great advantage of the technology is the following: it hones in on highly conserved cytosines, and second, it provides very deep sequencing depth. So if you have one cytosine, you can measure its methylation very, very accurately. Conversely, though, the drawback of the technology is that it only looks at a few 10,000 cytosines, whereas most species have tens of millions of cytosines. For example, humans have 28 million cytosines in their genome. So I assembled a mammalian methylation consortium. We are a group of almost 200 people to profile the tissues of many, many animals. So these tissues came from existing repositories. People had freezers full of stored tissues and DNA. And I'll start with a study where we analyzed the effect of a treatment known as rapamycin in marmosets. Marmosets are a type of monkey. And the question in the field was, well, does rapamycin rejuvenate these animals? And there's a lot of evidence that rapamycin could be beneficial. And so I was quite hopeful that we would see an effect. But in this study, in blood, we didn't see one, so I need to be honest about that. And it could be that rapamycin simply doesn't affect the methylation levels of blood samples. Maybe it has a benefit on other tissues, other organs. And that's a major insight of these studies I conduct when it comes to evaluating different treatments, that most treatments actually only work in a subset of cell types and tissues. So a treatment may work in skin, but not in blood, or vice versa. Now I come to so-called third generation clocks. What is meant by that? These are multi-species clocks. And what does that mean? Imagine you have a pig and humans, very different species. And you want to have one regression model, one set of cytosines to measure age in both of these different species. That's then the resulting clock is a multi-species clock that applies to pigs and humans. It uses highly conserved cytosines, the same regression model. That's a multi-species clock. And I'll start here showing you what I call a pan-primate clock. So pan-primate clocks apply, this particular clock truly applies to all primate species. In the middle, you see Homo sapiens, humans. So this model is accurate at a correlation 0.98. It's an extremely accurate clock in humans, but excitingly, it also applies to very distant primates, lemurs in Madagascar, for example, or gorillas, or rhesus macaque. Here's this human pig clock for relative age. I want to maybe briefly mention what we mean by relative age. Relative age is the ratio of your calendar age divided by the maximum lifespan of your species. In pigs, the maximum lifespan is 23 years. The oldest pig that has ever been recorded has reached an age of 23. The oldest human ever observed reached an age of 122. And so by forming this ratio, this relative age estimate, we align these species, admittedly in a very basic way. However, this alignment, although it's quite basic and naive, it does work. So, and that can be seen from the scatter plot, where I show you the methylation estimate of relative age on the y-axis versus the actual relative age. And here we have the green dots are humans, and the other dots represent tissue samples in bladder and blood and other tissues from a pig. And similarly, we've built these third generation clocks that apply to rats and also to humans. So again, the left panel shows you relative age on the x-axis, y-axis is a cross-validation estimate of relative age. I want to briefly mention that any scatter plot I show you today is based on cross-validation. It's always an unbiased evaluation as opposed to a so-called training set estimate. And anyways, you see that the orange dots very much align with the green dots. So really, when we use these dual species clocks, we measure aging in both of these very different species. And now comes the question, so why build such a clock? Well, the answer is that we want to increase the chance that research findings in a rat translate to those in humans. As you know, we have cured diseases in rodent models over and over, but then the corresponding treatments fail us in human clinical trials. And by looking at these highly conserved cytosines and using these third-generation clocks, we hope to have a biomarker that applies then to both and leads to insights that translate to humans. Here I show you another study where we developed, again, a relative age estimator for humans and sheep. And then we applied it to ear samples from sheep to evaluate castration. And the blue bar on the left shows you that castrated males were epigenetically younger than intact male sheep. And so that would suggest that a castration does have a rejuvenating effect on the ear samples. But that effect is probably tissue specific for ear or skin, because I evaluated castration also in cats, dogs, and horses, and never found an effect in blood samples. So blood methylation does not seem to relate to these hormonal changes. Here I show you our clocks for cattle, bovine clocks. And this study came from a study of oocytes. We were very interested whether oocytes age. And the insight was that whatever changes with age in these oocytes really doesn't change in blood. So as perhaps expected, oocyte aging is fundamentally different from blood aging. A lot of my collaborators are interested in conservation of species, endangered species, and to properly manage endangered species, you really need to know their ages, because you want to see whether suddenly there is an insufficient number of young animals in a population. So you need to find non-invasive ways of measuring age in wild animals. And therefore, we developed elephant clocks. So we have very accurate clocks for African elephants and Asian elephant. And for no good reason, I also built a human elephant clock. I cannot think of any utility of it beyond saying I wanted to emphasize a conserved biology. You know, there are cytosines that whose methylation increases with age in humans, and they are at the same location in elephants, at the same genes. Similarly, we have epigenetic clocks for cats and humans and cats. So we have clocks that are based on blood samples from cats. I have no doubt they also apply to saliva samples from cats. And so my pitch was always, if you want to measure the age of your cat that you may have received from a shelter, you can do it, but you can also add your own saliva sample. So we can measure not only the age of the cat, but also that of the human owner of the cat using the same human cat clock. So you can compete with your cat on the relative age measure, who is younger. Here I show you a study of 93 different dog breeds, a collaboration with Elaine Ostrander from the NCI. So we profiled 700 blood samples from 93 different dog breeds. This clock is very accurate, again, correlation 0.97. It applies to all dog breeds. But so this, let's be very clear, this is now a chronologic age estimator. Clearly, it would be much more exciting to have a predictor of a mortality risk for your dog. And in that paper, we already described a prototype of such a clock. We call it a predictor of average time to death. And it applies across different dog breeds. But in the discussion of our paper, and here as part of this conference, I want to emphasize we really need to validate this predictor in independent test data, where we focus on one single dog breed and need to see, similar to what we have in human grim age studies, we need retrospective data to see whether these predictors work and to what extent. But overall, I would say this is a current frontier in the field of companion animals to build second-generation clocks. Now I come to an animal that is famous in my community. My community is really aging research, rejuvenation research. The naked mole rat. The naked mole rat was at some point the animal of the year because it seems to exhibit what is known as negligible senescence. When you look at that animal, it doesn't seem to age. And the naked mole rat, despite its small size, can live up to 38 years, I think. So really long living species. And so people thought, well, maybe this is the one mammalian species that does not age. But our epigenetic clock studies clearly show that you can build very accurate clocks for the naked mole rat. So according to our methylation study, this naked mole rat, the naked mole rat ages like all other mammalian species. And we, of course, built also human naked mole rat clocks, illustrating, again, conserved biology. And that brings me to another point. Humans are a very long-lived species. We have an exceptionally long maximum lifespan for our taxonomic order. So in that sense, I would say the naked mole rat is not much different from humans. Again, coming to the topic of conservation efforts, Tasmanian devil was endangered. So, and we built epigenetic clocks for all marsupial species, kangaroos, Tasmanian devil, also opossums. I have to say that overall these clocks are not that accurate. I mean, you see correlations 0.9, 0.85. But anyway, I would say they're not as accurate as in eutherian species, which could be due to two reasons: biologic differences, but more likely the fact that our mammalian array contains more cytosines that work to eutherian species than for marsupial species. Now I come to a different application, bat species. Again, conservation efforts. So we analyzed wing punches, tiny, minute selections of skin from these bats, 700 samples from 26 different bat species. And here I illustrate that, yes, we have very accurate clocks for bats, and they are being used in conservation efforts. So let me come to the most challenging question. Can one build epigenetic clocks that actually apply to all mammalian species? Marsupials like kangaroos, eutherians, placental mammals, monotremata, is that possible? And here I show you that the answer is yes. So this scatter plot shows you on the right-hand side, in the right side, you see again relative age, a number between 0 and 1. And on the y-axis, you see the methylation estimate based on cross-validation. And look at this correlation, 0.95. So although we use here over 200 different mammalian species, different ages, different tissue types, we can measure age with one regression model, one formula. And the main application is really to demonstrate that on an epigenetic level, development and aging are quite conserved if you look at these conserved stretches of DNA. Here I show you how this universal pan-mammalian clock performs in humans, upper left, in mice, in dolphins, in dogs, in elephants, in bats. Now, such a clock is not as accurate as a species-specific clock. If you could look at the lower left, these are the same dog samples I mentioned before. So this universal clock is less accurate than the dog clock I showed you, but it's still remarkably accurate, correlation 0.94. So these clocks really usher in a new era of research. We can use the same epigenetic clock to measure aging in several species, mouse, rat, mini pig, sheep. And my hope is that this will facilitate the translation of treatments, not just from a rodent model to humans, but conversely, the opposite, from humans to your pets. Because imagine we have a wonderful treatment that rejuvenates the human owner of a companion animal. They will say, well, can't we tweak that treatment, or can we change it so that it also benefits my pet? And so maybe these types of biomarkers can be useful. I did not talk much about so-called marine mammals, but I want to tell you there must be by now a dozen publications where we and many others describe clocks for any marine mammal you can name, dolphins, different whales, and so on. For the biologists among you, you may ask, well, what hallmarks of aging relate to epigenetic clocks? Because I focus here on cytosine methylation, but there are many things that change with aging. And here I list several hallmarks that are well known. Start with cellular senescence, telomere length, genomic instability. These are well-known changes with aging, but surprisingly, they seem to be independent of epigenetic aging. I mean, they matter very much for aging, but not epigenetic aging, apparently. And conversely, these other hallmarks, so-called nutrient sensing, I mentioned obesity, for example, increases your epigenetic age, or high-fat diet, and or I mentioned conversely nutrients such as vegetables. So they all relate. Mitochondrial dysfunctions relates stem cell exhaustion and cell-cell communication. And this is a paper by Ken Raj, who looked at in vitro aging. So these are human in vitro studies. I'm coming towards the end, and I want to briefly mention another angle of the research in my lab. We, of course, are quite interested in the mystery of maximum lifespan. Why is it that a bowhead whale can live up to 211 years? Conversely, why is it that certain rodents, shrews, mice, rats, live short lifespans? Certain bats can live 40 years. So just this mystery of lifespan differences. And part of the answer is undoubtedly methylation. And I'll show you this. On the x-axis, you see the observed maximum lifespan. And the y-axis shows you a methylation-based estimate, again, a cross-validation estimate, where we built a multivariate predictor based on cytosines. Correlation 0.89, so it just illustrates strong effects. I notice we have some extra time, and I want to briefly discuss what I've learned about how to slow human aging. And here I show you a study where we studied the effect of obesity on human liver samples. And on in the upper panel, you see how epigenetic age relates to chronologic age in liver samples, just making the point that our clocks really do work in all nucleated cells, here liver cells. The bottom panel shows you body mass index in humans versus this age acceleration. And you see a correlation 0.42, and this is just the finding that, yes, obesity has a strong effect on the epigenetic age of the liver. The effect in blood, much weaker. It's still detectable, but much, much weaker. And I studied many human post-mortem tissues to see, well, what kind of conditions are associated with increased epigenetic age in different organs. And the top row shows you the punchline: hypertension. Hypertension, high blood pressure increases the epigenetic age of human fat tissue, kidney tissue, liver tissue, muscle tissue. But interestingly, I barely saw an effect in human blood, So again, illustrating that certain stress conditions affect certain cell types, but not others. And I want to talk about menopausal hormone therapy, which is risky. Many doctors advise against it. But we looked at keratinocytes, really buccal swab samples, and found that, yes, women who had taken menopausal hormone therapy exhibited a younger epigenetic age in their skin samples. Conversely, though, we didn't see any effect in human blood samples. I want to now talk about progeria. These are accelerated aging disorders. For me, the #1 genetic disorder that increases your epigenetic age is Down syndrome, trisomy 21. Then there's another condition known as Werner syndrome, or also known as adult progeria. We found that the blood samples of these individuals exhibited increased epigenetic age. And then there's another condition, so-called cocaine syndrome, where we also saw that. I briefly want to describe a study in mice where we again found that Down syndrome is associated with increased epigenetic age in mouse blood, cerebral cortex, and liver samples. And so this is an animal model of human Down syndrome. I want to come to another progeria that involves children. So this is now Hutchinson-Gilford progeria. And unfortunately, these children exhibit aging effects early on in life. And to my disappointment, our methylation clocks don't really find strong evidence of epigenetic aging. So it's disappointing because on a clinical level, clearly accelerated aging, but our, for example, the blood samples in these children don't reflect the disease. And I mention it to make a point. Not everything that has an adverse effect on your health can be measured using an epigenetic clocks. Epigenetic clocks are one biomarker out of several other biomarkers. Many other clinical biomarkers should be considered. For example, blood pressure, glucose levels, and 10 other markers for various organ dysfunction. I now want to come to a first-generation clock where we measure chronologic age in humans. Old topic, but a new application. We want to validate the age claims of centenarians. And this research was motivated by a recent controversy 2 years ago, where some researchers felt that Jeanne Calment, the oldest person who ever lived, conducted a fraud a fraud in order to avoid inheritance taxes. And so they cast doubt on her true age. So this then leads to a need for a molecular biomarker that allows us to verify age claims. And so we developed an epigenetic clock for that purpose. So here I show you again a scatter plot, but the most important part of it is the x-axis, the chronologic age. Notice that all of these blood samples and saliva samples come from centenarians, some of them maybe 116 years old, very old. And we developed the centenarian clock that is still remarkably accurate, median error less than 3 years, in order to verify age claims. So if somebody comes and says, oh, you know what, I'm 130 years old, we could analyze their blood and aim to corroborate or refute it. And so we and others, of course, study centenarians because these are wonderful examples of individuals who are highly robust to many forms of stress and insults. And we may be able to learn their secret. So to conclude my presentation, I mentioned epigenetic clocks have been linked to 4 hallmarks of aging: stem cell exhaustion, nutrient sensing, mitochondrial dysfunction, intercellular communication. Epigenetic age is very distinct from telomere length and senescence. I've shown you that the cytosine methylation levels allow you to build pan-mammalian and pan-tissue clocks. I've illustrated that cytosine methylation even allows you to estimate the maximum lifespan of a species. I've shown you methylation allows you to estimate human mortality risk. We have epigenetic clocks for most model systems, including dogs, pigs, primates, and rodents. I illustrated epigenetic clocks are being used in clinical and preclinical studies of anti-aging interventions. And I have started a non-profit foundation, the Epigenetic Clock Foundation, that offers epigenetic clock testing to the research community and also commercial partners. Really, we want to promote the rigorous use of epigenetic clocks in research. Thank you so much.